Hi everyone, welcome to our scientific webinar series. As you can see, we've got two speakers today. We're fortunate to be joined by Lysan and Maria from Lilly. As you can see, we've done things a bit differently this time in terms of opening up to a bigger audience than usual, and I hope that'll be well received, mainly because it's quite an interesting and case study, which we're really glad that Maria and Lysan have agreed to talk on. And if you just want to go on to the next slide, I'll introduce our two speakers. I can't see the next slide yet.
There's just a little bit of a delay.
Thank you. Well, while that's loading up, I'll introduce Lysan, who is a principal research scientist in global pharmacokinetics, pharmacodynamics, and pharmacometrics. So she wears lots of hats by the sounds of it, and a busy person in Eli Lilly. She is joining us from Singapore. So again, we're really grateful to her for joining at this hour to present her work. A lot of you maybe in the SimSip community would know Maria well. She's a principal research scientist in and specializes in ADME and obviously in PBPK as well. And today, both of them are going to be presenting on this case studies involving Dulaglutide and its mediated gastric emptying delay on drug exposure. So I'm not going to talk anymore. I'll hand over to you both, and thanks again.
Thank you. Okay. Good morning, good afternoon, and good evening to the audience. It's our pleasure here today to share with you some of the work that Maria and I and a group of our Lilly colleagues worked on, looking at predicting the effect of Dulaglutide-mediated gastric emptying on drug exposures. And in terms of drug exposures, we are actually referring to the exposures of a group of drugs commonly taken together when patients are on dulaglutide therapy. So just a little bit more on the background of dulaglutide. Some of you may be more familiar with the trade name of dulaglutide, and that's Trulicity. Dulaglutide is actually once weekly what we call a GLP-1 receptor agonist. GLP stands for glucagon-like peptide 1, and it is administered subcutaneously for purposes of improving glycemic control in adults with type 2 diabetes. Dulaglutide was first approved in September 2014 in the US at 2 doses, 0.75 milligrams and 1.5 milligrams, both given once a week. 2 additional doses were approved in September 2020, so that's more recently. And the indications for these 2 additional doses were for patients who need additional glycemic control. So basically, they haven't quite met their glycemic control targets. The focus today will be on the development of 3 and 4.5 milligrams and how we actually took a modeling approach to evaluate drug-drug interactions. But throughout the development life cycle of these additional doses of dulaglutide, we took a model-informed drug development to help us answer a lot of questions, actually. And these models were applied to facilitate and accelerate the decision-making in terms of picking the 2 doses itself, 3 and 4.5 milligrams. We also have to optimize the dose escalation regimen to getting to these two additional doses, because as a lot of you may be aware of, GLP-1 receptor agonists, they do actually have pretty well-known gastrointestinal side effects such as nausea, vomiting, and sometimes even diarrhea. But it is known that if you were to dose escalate gradually, you will be able to mitigate quite a bit of this GI side effects. And we optimize the dose regimen through a series of modeling to actually come up with a regimen of a 4-weekly dose escalation interval. So basically, you work through the dose range from 0.75 to 4.5 using a 4-weekly dose escalations. The highlight of today, of course, is to evaluate drug-drug interactions of dulaglutide with some commonly co-administered oral medications. And this is done via a combination of POP PK modeling and PB PK models. Together, these models actually help to support the label language around drug-drug interaction, as well as dosing around the special populations that were in our label. And the reason why we are able to utilize a model-informed drug development approach here is because there's actually a lot of data already available in the original dulaglutide submission. So we leverage on this vast amount of data and use that to build our models to take this MIDD approach for you know, the various decisions that were driven with model-informed drug development. I'm just going to move to the next slide. Oops. Okay, so a little bit of the background about GLP-1 receptor agonists, because some of you may be wondering, for a drug like dulaglutide and many of the other GLP-1 receptor agonists that are given subcutaneously, why are we concerned with drug-drug interactions with drugs that are taken orally? And that's because GLP-1 has got several pharmacological effects. Centrally, they actually can increase nausea, vomiting, and also the feeling of fullness or satiety, which is neurally mediated. On the stomach, it actually GLP-1s can actually slow gastric motility, thereby causing an increase in gastric emptying time. And that's why any oral medication that's taken along with an injectable, in this case, such as a GLP-1, there's still going to be an interaction in terms of the a delay in the gastric emptying time of the oral medication. In addition to actually slowing down the transit of oral medications, basically the transit of even meal contents through the stomach will be slowed down. And that's actually beneficial in certain ways, because your postprandial glucose excursion will also be slightly depressed. And that actually works out well for type 2 diabetics. And of course, one of the other more important effects of GLP-1 receptor agonists is directly on the pancreas. And they actually mediate a glucose-dependent increase in insulin secretion. So let's go back and focus on a gastric emptying time. And a method that's commonly used to understand gastric emptying time for GLP-1 receptor agonists is actually via acetaminophen PK profiles. And I'll talk a little bit more about that in the next slide. So what we have done here in terms of characterizing the gastric emptying delay of dulaglutide is to actually administer a dose of oral acetaminophen to our patients or subjects, and then look at the PK profile or the systemic concentrations of acetaminophen after the oral dose. The reason why acetaminophen is a good marker for gastric emptying is because it is actually very poorly absorbed in the stomach, but it's absorbed very rapidly once it is being emptied into the small intestine. So if there's a delay in gastric emptying, it would actually show up in the systemic disposition of acetaminophen. Bearing in mind actually that the gold standard for studying gastric emptying or transit times is actually via scintigraphy, but scintigraphy is an expensive and requires imaging capabilities in order to conduct those studies. So basically, you have to feed the subject a radio-labeled meal and then track the residual amount of radio-labeled activity that's left in the stomach over a period of time in order to characterize the transition of the meal in the stomach. And there's lots of data out there that shows that acetaminophen PK is a very good surrogate marker for gastric emptying time, and the results correlates closely with scintigraphy data. So with the PK data of acetaminophen on hand, what we did was we built a population PK model using nonlinear effects, nonlinear mixed effects modeling, and most of you, I think, in this audience would be familiar, and that's done via non-MEM. What we would then need to do is to implement this delay in gastric emptying time into the PBPK models, which are conducted on SimSip. And as you know, non-MEM and SimSip, they operate on two different platforms. So what we need to do is to convert the parameter around delay gastric emptying into a common parameter that can be integrated into the individual PBPK models of commonly co-administered oral medications. And a list of these medications are shown on the right-hand corner here. And they are generally medicines that are commonly taken by type 2 diabetic patients or medications that have a narrow therapeutic index. Okay, I'm going to move to the next slide. I'll talk a little bit more about this idea of how we convert things into a common parameter so that we can make the two different platforms talk to each other. So in this figure with the yellow heading, what you see is a series of green little boxes here, and this is actually the POP-PK model of acetaminophen or paracetamol. And you can see that it tracks the transition of acetaminophen from the stomach into the small intestine. And what you have here is a term FPL, and that's actually the first pass effect that is lost from the small intestine. And subsequently, what you have here is, post-absorption is the typical central and peripheral compartments of distribution for acetaminophen. This term here that I've circled is actually a first-order rate constant, signifying the gastric emptying rate from the stomach into the small intestine. And overlay on top of it is actually a POP-PK model for dulaglutide, and those are the blue boxes. And this influence of dulaglutide actually occurs in a dose-dependent manner on this gastric emptying rate constant for acetaminophen. So taking this gastric emptying rate constant over a wide range of dulaglutide doses or exposures, you come to this equation at the bottom where we convert this gastric emptying rate constant into a time factor. And so if you take this and imagine it like a half-life and you use a natural log of 2 divided by the rate constant, you get a time factor, which is actually the time to 50% of acetaminophen gastric emptying. And with this time to 50% of acetaminophen gastric emptying, we come to this blue box here in the middle, this figure here. We simulated the gastric emptying time over a wide range of dulaglutide exposures that corresponds to the doses of dulaglutide that we were interested in, and that's 0.75, 1.5, 3, and 4.5. And what you get here is a non-linear curve. The reason why we're getting two curved lines here is because the data set going into the acetaminophen model actually comes from two different studies, one in healthy volunteers and one in type 2 diabetics. And you can see that the curves are actually different. Type 2 diabetics actually have a faster gastric emptying time for acetaminophen compared to healthy subjects. And taking this time to 50% gastric emptying, we now have to find a way to implement this into a model, and a time parameter is difficult to implement this into the PBPK model. So this work was actually done with one of our pharmacometrician colleagues, Karen Schneck. And what was done here is we converted this into a fold change or a ratio. So each of these colored dots, we divided it by the baseline, the gray dot here, where there is no dulaglutide on board. And we got a fold change in gastric emptying time for each exposure corresponding to a dose of dulaglutide. And we then converted this to what's in the green box here, where we plotted the fold change in gastric emptying time versus, again, a range of dulaglutide exposures. What's interesting, however, is that when we did this conversion, we realized that these two curves actually converged into one. So the fold change actually was the same for type 2 diabetics and healthy subjects, despite the difference in the actual gastric emptying times. And therefore, this fold change was dose dependent, and taking the 4.5 milligram dose, where the fold change was close to about 5-fold, which is probably the worst case scenario, we took this value and then implemented this into the gastric mean residence time in the individual PBPK models of the commonly administered oral medications. So this slide here shows you the data going into the model building of the acetaminophen POP-PK model. So as I've mentioned, we had two studies, a 5-week study where dulaglutide was given weekly over 5 weeks in type 2 diabetics, and a 4-week study in healthy subjects. Dulaglutide dose range in the type 2 diabetic study ranged from 0.05 to 8 milligrams and was more than adequate to cover for the dose range that we're interested in, which is 0.75 to 4.5 milligrams. In the healthy subjects, the dose range was between 1 and 3 milligrams. And acetaminophen was given on at baseline and on subsequent days. Typically day 3 or day 2, which is about 2 days after a dose of dulaglutide, and that typically signifies the peak concentration or the Cmax for dulaglutide. And then acetaminophen was sampled over between 12 to 24 hours, and that's more than adequate to fully characterize the PK profile of acetaminophen. Okay, so this is, sorry, slide jumped. Okay, this is an illustrative example of one interesting phenomenon on top of the effect of dulaglutide on gastric emptying being a dose-dependent one. This is an additional phenomenon that we had observed. So taking a typical dose of dulaglutide, 1.5 milligram, which is the most commonly prescribed dose, subjects patients were given acetaminophen doses of 1 gram at baseline, so that's day -1. At day 2, right after a dose of the first dose of dulaglutide, somewhere around its peak, and then at day 23, which is around the peak of dulaglutide after 4 doses. And acetaminophen PK was plotted out here in this profile. At baseline, what you see is the blue curve. At day 2, after a first single dose of dulaglutide, you see this green line, and that's clearly a little bit more depressed than the blue curve, showing the effect of delayed gastric emptying on the acetaminophen profile. However, following 4 doses of dulaglutide on day 23, you see this red line here. This red PK profile of acetaminophen has almost gone back to what it was at baseline. So this is the additional observation that besides being dose dependent, the effect of dulaglutide on acetaminophen PK gastric emptying is largest after the first dose, and it diminishes over time with repeated dosing. So it is therefore appropriate that when we are evaluating the gastric emptying delay effect of dulaglutide, that we took the highest dose, the fold change at the highest dose of 4.5 milligrams, and also took the fold change at at around day 2, which is the fold change after a first dose, because that's the worst case scenario. One thing to bear in mind, though, is that dulaglutide at 3 and 4.5 milligrams are not recommended as starting doses. They are, in fact, doses that you could give to the patients if they need additional glycemic control. So we would expect that most of these patients would have gone through several weeks or longer of dulaglutide treatment, and therefore some of this delayed gastric emptying effect may actually have diminished. So taking the fold change at 4.5 milligrams after a single dose was actually really the worst possible scenario. And this slide here, what I'm trying to show you is that the population PK model of acetaminophen actually described the data very well, including the delay in gastric emptying, and you can see more depressed curves here. This happens over a wide range of dulaglutide doses in the type 2 DM study. The black circles are the observed acetaminophen concentrations. The bands are the model predictions and the 90% prediction intervals. So you can see most of the observations actually sit within the model predictions. Same for healthy subjects. So in a case at baseline where there's no dulaglutide on board, the model described the data well, and the model continued to describe the data well with dulaglutide on board. So this model that we had for acetaminophen can describe gastric emptying delay under various conditions, so with or without dulaglutide on board, and over a range of dulaglutide doses that covers the doses that we're interested in investigating. And we can also do this in healthy subjects and type 2 diabetics. So the model is pretty robust for generating the fold change that we're interested in. So by taking this novel integration of traditional POP-PK modeling along with PBPK modeling of individual conmats, what we have managed to do is that you can see in our label, SMPC, that's our European label, we managed to actually conduct our drug-drug interaction evaluations up to the 4.5 milligram dose and conclude that it's not clinically relevant, with the label actually calling out this evaluation being justified by a PBPK modeling. And in the US label, in our USPI, we have a forest plot here, and you can see that the Cmax and AUC, and I'm sorry that yeah, it's a little bit small, might be a bit difficult for you to see, but they're generally on the line of unity, which is the reference where there is no dulaglutide on board. And Maria will probably speak to you a little bit more about the details on the individual PBPK models. And all these, we actually managed to achieve them without running any real or additional clinical drug-drug interaction studies at the 2 additional doses. They were all achieved via modeling approaches. Maria, all yours now.
Thank you, Lyston. Can you hear me?
Yeah.
Great, thank you. So as Lyston just stated, multiple drug interaction clinical studies were run with the lower doses of 1 and 1.5 milligram dulaglutide after a single dose to assess the impact of the delayed gastric emptying on multiple small molecules. And these were the 9 molecules we evaluated. And you can see that the effect of gastric emptying delay does not affect all molecules the same. For some of these molecules, the AUC and the Cmax don't change at all, or change very little. For others, the Cmax and or the AUC decrease, so are below that 1. And for others, the Cmax and the AUC actually increase. So it's not that all molecules are affected the same. You have to kind of look and understand the properties of these molecules to be able to understand and predict the effect of gastric emptying delay on these small molecules. So our first approach was to look at this in a qualitative way. The approach we took here was to analyze and devise these drugs based on the categories from the Biopharmaceutics classification system. And the BCS is a framework established by Gordon Amidon and collaborators at the University of Michigan to classify drugs based on their permeability and aqueous solubility. This classification system has 4 categories. Class 1 are drugs with high permeability and high solubility. Class 2 are compounds that have high permeability but low solubility. Class 3 are compounds that have high solubility and low permeability. And Class 4 are those who have both low solubility and then low permeability. So what happens with gastric emptying delays for BCS Class 1 compounds? So these compounds, as I just mentioned, have high solubility, high permeability, and are well absorbed. And because of these compounds can get absorbed as soon as they leave the stomach and get into the intestine, the absorption rate for these compounds, or compounds in this category, could be limited and affected by changes in gastric emptying time. Therefore, in a qualitative way, you would think that this, or you would see that a delay in gastric emptying for this would affect these compounds, and it would increase the Tmax, it would decrease the Cmax, but usually will not have an effect on AUC. Drugs that belong to the BCS class 3 category, which again have high solubility but low permeability, are not really expected to be affected. And it's because the rate limiting step for the absorption for these molecules is the permeation through the intestinal wall. Therefore, when you change gastric emptying time, you will not really see an effect on the absorption. And this is why you would really not expect a change in Cmax or AUC, and you might see just a delay in the Tmax. For BCS class 2 compounds that have low solubility and high permeability, and are acidic compounds, we really don't expect an effect. These compounds are not really going to get absorbed right after they leave the stomach, so we won't see really an effect on Cmax or AUC, and you might see just a delay in AUC. And something similar happens to the compounds in the for acidic compounds in the BCS class 4 category. Sorry, that jump. Now, for basic compounds, you might see for the BCS class 2 and BCS class 4, you might see that a delay in gastric emptying might allow for a little bit more of the dissolution of the product in the stomach, because they're going to sit for a longer time in the stomach. And that might cause an increase in Cmax and AUC for BCS class 2, but not really it's not really going to have a big impact for BCS class 4 compounds, since they're still do not have low permeability. Okay, so that was kind of our qualitative assessment, and it is very helpful because it kind of guides you and to understand like what should happen, and on a qualitative way, what should happen. But in order for us to predict the actual changes, the magnitude of these changes, and the impact of the gastric emptying delay on the exposure of these drugs, we have to use a quantitative model method like PBPK model. And PBPK model, as you saw before, coupled or informed by the results of a POC-PK model. So this is, in this case, we chose SimZip as for the software for the PBPK models, and because we're looking at absorption, we had to make sure that the advanced dissolution, absorption, and metabolism model, also called ADAM model, was used for all the PBPK models for the small molecules. And we do that. We use PBPK model because it has an independent set of parameters. So we have the drug parameters, we have the physiological parameters, and the population parameters. And we can actually alter one of these parameters without altering the others, and this is what, and in this case, we alter gastric mean residence time to be able to reproduce the effect of dulaglutide. Okay. So for our for our PBPK models, we either develop or have our own develop our own PBPK models. We modified some literature PBPK models that we found, and we also use some of the SimCip PBPK models available in the SimCip library and modify them to incorporate the ADA model or the absorption mechanistic model. And as Lysan mentioned, the key parameter for this project is, in SimCip, is called the gastric mean residence time. And our assumptions were that at baseline, the fasted, the gastric mean residence time was 0.27 hours, and the fed was 1.18 hours. So this was without dulaglutide. And the way, to calculate the gastric mean residence time with the different doses of dulaglutide was just by multiplying, as Lysan mentioned, the fold change in gastric emptying from that POC-PK model or POC-PK-PD model with the baseline mean residence time in SimCip. And not only we assume the same fold change for the diabetes population and the healthy volunteer population, like as Lysan showed, but we also assumed there was the same fold change in the fed and fasted state. Because we had some studies that were run in the fed state. And since, and for this fed state models, we had to use the segregated transit time model to allow for gastric mean residence times that are above 4 hours. The two key input or key sets of inputs for these models were the permeability input and the solubility of this or dissolution inputs. So for permeability, we search in the literature for a measure human effective permeability values. So measured in human healthy volunteers, usually with the Lucky Gut method. If those were not available, we use our in vitro apparent permeability values measured MVCK cells and translated this using in vitro in vivo correlation to in vivo human effective permeability values. For the formulation characteristics, the solubility or dissolution characteristics, for the BCS class 1 and class 3 compounds, since they're really not, they have usually high solubility, we assume a solution without precipitation for these molecules. For the other molecules, we had either immediate release, controlled release, or solution with precipitation. And in some cases, we prefer to use dissolution values, dissolution profiles, published dissolution profiles, instead of inputting the solubility values. And these were the 9 molecules that, again, that we modeled for this exercise. So let's start with the verification of acetaminophen, of the acetaminophen model. For this molecule, we used a modified model published by Zhang and collaborators. They had used the model. This model originally had a first order absorption component, so a Ka and an Fa, and we modified it to incorporate the ADA model, and we kept a solution because in this study, in this PBPK model, they built these models for both the solution and the elixir, and we use our own measure apparent, so in vitro permeability, and convert that to a human in vivo P effective, and as expected, the fraction absorbed acetaminophen was high, was 1. So this is how the performance of the model. So the model actually can reproduce the effect of, the pharmacokinetics of acetaminophen by itself, and also which are shown. The concentration time profile is shown on the graph on the top left. And you can see in this graph, and I'm going to present a few of these graphs, and usually, these graphs have the line represents the mean predicted concentration time profile. The dashed lines are the 5th and 95th percentile, and the dots with the error bars represent the observed concentration time profile in the individuals in this study. So this is the data from the dulaglutide study. On the left, we have the placebo, and on the right, we have with 1 milligram dulaglutide. And the model was able to capture the pharmacokinetics of acetaminophen in the placebo and the dulaglutide arm. And it was also able to capture the change in Cmax and AUC of acetaminophen with 1 milligram dulaglutide. So these are expected. This delay in Tmax, decrease in Cmax, and no change in AUC is expected for a BCS class 1 compound, as I mentioned before. And on the bottom graph, you see a sensitivity analysis of the Tmax. So with increasing gastric mean residence time, you expect a larger Tmax. With an increase in mean gastric residence time on the x-axis, you can see in the middle graph, you can see that the Cmax goes down. And on the right graph, you can see that the acetaminophen AUC does not really change much with changes in gastric emptying, or as expected with a BCS class 1 compound. Okay, so the other example of a BCS class compound is warfarin. For warfarin, we modified the model available in Simcyp, and we enabled the atom model, and we model it as a solution without precipitation, and use our own in-house measure apparent permeability to predict the human P effective. And the fraction absorbed, due to the high permeability, is predicted to be really high, close to 1 or 1. And this is how the model performs. So the model is able, again, to reproduce the pharmacokinetics of warfarin in the placebo state and with 1.5 milligram dulaglutide, and can also reproduce the change, the Cmax ratio and the AUC ratio for warfarin in the presence of 1.5 milligram dulaglutide. So here's, again, another sensitivity analysis. This one is for warfarin, and what you see is similar to what we saw with acetaminophen, because warfarin has high solubility, high permeability, and a gastric emptying delay is going to limit its absorption, or the rate of absorption. So the bottom left graph shows the Tmax on the y-axis and the gastric mean residence time on the x-axis, and you can see that the Tmax is sensitive to that change in mean residence time and increases with increasing mean residence time. And the Cmax, on the other hand, as expected, is going to decrease with increases in mean residence time, and the AUC is not predicted to change with changes in mean residence time. Okay, so those were the two examples, some of the two examples we had for BCS class 1 compounds. So now let's switch classes to the BCS class 3 compounds. The first compound in this class that we had was Lystenopril. So for Lystenopril, we built developed our own model, and based on literature data, reported clinical studies, and we use, again, the advanced dissolution, absorption, and metabolism model. And we used in the model a formulation that was solution without precipitation, which, as I said before, this has high solubility and compounds, so we don't expect really precipitation in the gastrointestinal tract. The reported viabilities range between 0.2 and 0.6 in the literature. And we also had two options for permeability. We had a reported permeability by Hans Lennernath, and this is a P-effective permeability measure in the human jejunum, and I believe this was with the lucky gut. And this permeability predicted a fraction of absorb of 0.6. We also had an in vitro in-house measure permeability of 0.1.5 times that would predict a human PF of 0.15 times 10 to the minus 4 centimeters per second. And this predicted a fraction absorbed of 0.4. So both these values, the human measured and the predicted in vitro, were within the range of the reported bioavailabilities. So one of the things we did to kind of figure out which permeability to use for the final model was to run a few simulations with different permeabilities and see how well they predicted the observed data. So the graph on the top left is the Lystenopril plasma concentration on the y-axis, and on the x-axis is the time. And the blue line, sorry, let's start with this. The dots, those are squares. The solid squares represent the observed plasma concentration time points for the dulaglutide study in the placebo arm or in the placebo subjects, and the error bars are the standard deviations. Now, the blue line on the bottom is the model or the simulated concentration time profile using the in vitro apparent permeability to predict the P effective. The red graph on the top is the simulated concentration time profile with the in vivo P effective. And the green line was a fitted concentration time profile. I wanted to fit and see how off, we would be from this to what would the permeability be if we use the concentration time profile. So they are quite close to each other, and the overall impact of this low permeability on the model and on the AUC and Cmax ratio was minimal. So we decided to kind of stick with a mechanistic approach of not fitting parameters from plasma concentration profiles to avoid identifiability issues. So we decided to go forward with the P effective that was measured and reported by Lennernas in 2007, and continued with that for our model. And so, as I said before, it's not really perfect. It doesn't really match the Tmax, but the Cmax and the AUCs are quite well predicted, and in the placebo and in the dulaglutide arm. And most importantly, the model can reproduce the lack of effect of gastric emptying delay. And as I said before, this is as expected, given that we don't really, that the absorption of these compounds is not really going to be limited by gastric emptying. It is going to be limited by the permeation in the small intestinal wall, through the small intestinal wall. So again, here you can see the results of the sensitivity analysis for Lystenopril, and this is what we would expect with other BCS class 3 compounds, where when the gastric mean residence time changes or increases, we see minimal change in the Tmax, no change in Cmax, and no change in AUC. So the other compound we had in the BCS class 3 category was sitagliptin. For this, again, we built our own models based on published data, and the model, again, can reproduce the pharmacokinetics and the placebo arm with 1.5 milligram dulaglutide. And again, the model reproduces the observations that there's no really an impact on the pharmacokinetics, on the Cmax or the AUC of sitagliptin. So as usual, in science, we come up with the things that really don't match what we think are happening. And this is kind of what happened with metoprolol and atorvastatin for this project. Metoprolol atorvastatin is a BCS class 2 compound, and we really were not expecting a big impact or an impact of gastric emptying delay on the Cmax. And we saw that there was around 70% reduction in Cmax for atorvastatin. And for metoprolol, which was given as a controlled release formulation, we actually saw an increase in the AUC and the Cmax. So all the other compounds work, the predictions work well with our initial assumptions of delay in gastric emptying, except for the models, the initial models for these two compounds. So if you're an optimist like me, you look at it and say, okay, here's a learning opportunity, and let's see what's happening with this molecules. So we I'm going to start with BCS class 2, atorvastatin. And this is a compound that Bridget Morse, a colleague of mine, did a lot of research on and a lot of experiments trying to figure out why that Cmax ratio was so much lower than for the other molecules. And it is quite complicated, the disposition of atorvastatin. And she also finds out that atorvastatin is really unstable in gastric fluid. So on the left hand is a picture from her publication, and the reference for the publication is there on the bottom of the slide. She shows that with time, the atorvastatin acid converts to the lactone in the low pH, and this is what you would expect happens in humans in the stomach. So the figure on the right shows the complicated, as I say, atorvastatin has quite a complicated disposition. So it shows the disposition of atorvastatin and some of its metabolites. And so atorvastatin is administered as the acid, and then in the stomach, it gets converted to the lactone. And then both the lactone and the acid are absorbed and metabolized in the liver. And then in the systemic circulation, you see a conversion of atorvastatin lactone back to atorvastatin. So knowing the disposition of atorvastatin and knowing that atorvastatin is unstable in the stomach, Bridget proposed that the delay in gastric emptying is causing a bigger conversion of atorvastatin to atorvastatin lactone, and therefore we see a decrease in the atorvastatin Cmax. Now, the interesting part is that we don't really see a change, significant change in the AUC ratio of atorvastatin, and that is again because once the lactone is absorbed, it converts in the systemic circulation, it converts back into atorvastatin. So it's quite interesting. I recommend you go and read her publication. And she also was able to reproduce what happens in the placebo concentration time profiles on the top, and with dulaglutide, the concentration time profiles on the bottom. And she was able to reproduce the changes in both AUC and Cmax for atorvastatin, for orthohydroxyatorvastatin, which is one of the active metabolites, and for the total species. So again, I recommend you read the paper. It's quite interesting, and it's she's a really good writer. So the next case is Metoprol. As I mentioned before, Metoprol was administered as a controlled release formulation. And for this model, we use the SimSip model and enable the mechanistic absorption part. And we input the permeability based on the measure values in humans from, again, Leonard's lab in Uppsala. And they measure the different segments. They measure the permeability in the jejunum, the ileum, and colon. And that was key for this project because we knew that Metoprol was being released all along the GI tract, and we really needed to have a good handle on what the permeability was in all these segments. The other key input were the dissolution. For this, we use dissolution time profiles at different pHs. Here I'm showing at pH of 1.2. We also had 4.5 and 6.5, but the dissolution is not really affected by the pH. So the model was really not able to reproduce the changes in intestinal, and the AUC and the Cmax when we were just changing the gastric emptying time. We had observations of a Cmax ratio of 1.32 and an AUC ratio of 1.19, and the predictions were a little bit above 1.04, but we were really not able to capture this increase in both AUC and Cmax. So while kind of looking at the literature, I found this publication and from Makatani and collaborators in 2017, and they show that liraglutide, which is another GLP-1 receptor agonist, decreases gastrointestinal tract motility, delays gastric emptying, and also slows small intestinal transit time. So this is not something we know happens in dulaglutide, but this was our kind of like one of our hypotheses. Okay, perhaps this could be happening to dulaglutide. In this study, in the Nakatani study, they use a pill cam. So a pill cam is a wireless motility capsule that has a camera and can record the pH, the pressure, and the temperature throughout the gastrointestinal tract when humans swallow it. And that also, because it has a camera, allows you to identify where the pill is located. So based on the results of this study, where they showed that the stomach changes 2.5-fold for patients with diabetes, and the small intestine changes a little bit less, so it's 1.8-fold. We use that ratio, that 2.5 to 1.8, to then predict what could happen with dulaglutide if this is what would happen with dulaglutide. And why is this important? Well, because if the small intestinal transit time is also being delayed, so the metoprol stays in the small intestine for longer, then it has a higher chance of being absorbed due to its low dissolution, because it's a controlled release formulation. So we use that ratio, that this 1.4, the 2.5 to 1.8, that 1.4 ratio, to then change in our model the small intestinal transit time, and then the model was actually able to reproduce what we saw for metoprolol when given as Metoprol XL, the controlled release metoprolol. So we don't have data for dulaglutide, but we do think that of all the hypotheses we have, this is kind of the one that makes the most sense. And not only we've seen that delay in gastric and small intestinal transit time, or that slowing of the small intestinal transit time with liraglutide, early on papers, when people were investigating the GLP-1 receptor agonist, they've also shown that in humans and primates, the small intestine motility decreases. So it proves to be quite a possible hypothesis. So after we built the models and submitted to the regulatory agencies, we got, as you could imagine, a lot of feedback. So they really wanted us to do more platform and model verification. They asked us to show dose ranging studies, single and multiple dose studies, see how the models predicted dose ranging studies, single and multiple dose studies, drug interaction studies. They also asked for us to predict a food effect, and we said that we really couldn't. Predicting food effect is very complicated, and we couldn't do that in all cases. But for the BCS class 1 compounds, we could, because one of basically the parameter that affects the Cmax and the AUC for this BCS class 1 compounds that have high bioavailability tends to be the changes in gastric emptying. So we did that only for those BCS class 1 compounds. And they also asked us to justify our assumptions of the mean residence, gastric mean residence time, and that we also had to justify the structure of the ADA model. So in collaboration with SimCip, with their help and the work of a lot of people, we are able to do the model and platform verification during the during the review process. And so this is a little summary of what we did. So as Lysan showed of the whole project, we start with estimating gastric emptying delayed with acetaminophen. We use a PK/PD or PubPK model in non-mem to get that change in gastric emptying. We input that into SimCip. We verify with the low doses of dulaglutide, and then we predicted with what would happen with the high doses of dulaglutide. And as Sam told you, this was successful, and we were able to use this for label language. And again, as I said, lots of people at Lilly work in this project. It was a really integrated effort. from many different areas in the company. And in conclusion, a combination of PK/PD and PBPK modeling approaches can be used to quantitatively predict the effect of delayed gastric emptying by dulaglutide. We had 9 clinical studies were replaced with simulations, and the conclusions were incorporated both in the US and the EU label. Again, we characterized that gastric emptying delayed using acetaminophen and PK/PD modeling, and we had to, in some cases, incorporate other mechanisms like changes in small intestinal transit time, or we realized that we need to focus and understand the stability of molecules in the stomach, like in the case of atorvastatin. And as I just mentioned, regulatory agencies ask for tremendous amounts of verification of model and platform verification in order to allow us to use this for the label. And I am done, and I would like to thank all the people that work in this project, and we were happy to take your questions.
Thanks. Thanks to you both. That was really great, and it was really nice to see POP-PK and PBPK kind of working together in a really positive way there, using them both. There are some questions that come in online. Just for people who are online who want to ask questions, there is a kind of an entry box that you can use for asking questions if you're interested. And if we don't get time to answer them now, we'll follow up with them in email. The first question, I think, Lysan, it's more appropriate that you take this one, but it was around how the baseline in gastric emptying time at dulaglutide dose 0 in healthy and diabetics was obtained. Some of the literature suggests that gastric emptying time is prolonged in diabetes compared to healthy due to gastroparesis, while the baseline you presented shows diabetes had a faster emptying than healthy. I guess the follow-up question was, can you share the source of baseline data? But yeah, I'll leave you to answer.
Sure. Yeah, so the baseline was established prior to the dosing of dulaglutide. So we basically fed the patients or the subjects a dose of acetaminophen, and then took the acetaminophen systemic PK profile and used that as an establishment of the baseline gastric emptying rate. Some of this data that I've shown, actually you can find it in a ACOP 2018 poster. It is out in the public domain. In terms of gastroparesis slowing gastric emptying time, I'm not an expert in gastroparesis, but I do believe that that occurs when the patients are chronically hyperglycemic, badly controlled, to the point where they actually get vagal damage. So at least in our study, that's not what we have seen in our patients, and that's lots of studies out there that also showed gastric emptying time is actually faster in type 2 diabetics. But I think there are many factors to that, so it depends on how well controlled they are, whether they are in a hyper or hypoglycemic state, and I guess also depends on whether you know, what kind of gastric content there is, so whether they are fed or fasted. So it depends on the condition in which I guess the gastric emptying test was conducted. I hope that answers the question.
Yep, that sounds clear to me. Thank you. In terms of the concentrations, there was a question about unbound drug concentrations or total concentrations that you were considering.
Yeah, I can answer that. Yes. So we don't expect a change in gastric emptying delay to change the fraction unbound or the intrinsic clearances. So we wouldn't really see a change in unbound concentrations. We presented was total concentrations. Yeah.
Yeah, that makes sense. So one of the follow-up questions, actually, Maria, you presented on the simulations. Did you run them in diabetes population as well, or was it all healthy?
No, we ran it in the healthy volunteers, yes. A lot of the clinical pharmacology studies were actually run in healthy volunteers. So we were trying to reproduce those studies, so that's why.
Okay. I'll just take a couple more. There's quite a few questions that come in, so obviously we'll follow up with them in email, just as I realize it's top of the hour. There's a question on effective permeability. Was it assumed to be consistent across the intestine, or did you use kind of the regional effects that you can use with the MECPEF model or something like that?
Yeah, so when we started this project, the MECPEF model was not available in this version. It took a long time to do this project. So the first molecules we didn't have the MECPEF model. Now for the model, now when we started version 17 and the MECPEF model was available, or whatever version that was, we did use it, except for the Metoprol case, where we actually had measured permeability in each of the segments based on the Leonard publication.
Yeah. Okay. So yeah, there's quite a few extra ones. Was dulaglutide evaluated in the clinic with staggered dosing to see if the time course of the change in gastric emptying, see that effect? And then I guess that's Lysan might be able to answer that one.
Yeah, so the short answer is no, because we evaluated the gastric emptying effect of the higher doses based on data that was in a study where the dulaglutide dose was given at a wide dose range, but not staggered. So at pretty high doses in type 2 diabetics.
Okay.
And certainly, maybe just a little bit more. And certainly the extent of delayed gastric emptying under those circumstances is actually going to be worse than when it is gradually dose escalated to the additional doses, where you give time for attenuation of this effect to occur.
Okay, thanks. I guess the follow-up on the flip side was in the modeling. So Maria, in terms of the gastric emptying delay change on the population value, or was it handled dynamically? I think I know the answer to this, but I'll let you answer.
So the question is, how did we put the gastric emptying?
Yeah, so how did you input the change essentially to the gastric emptying delay?
Yeah, so we took the PK/PD number and we made sure that we converted from a half-life to a mean residence time. So we did that conversion, and then we put it in SimCip in the in the input. So for each dose, we had to run a simulation. So it was kind of like one simulation per different gastric emptying number.
Yeah.
Manually, yes.
Yeah, maybe that's something to vote on in the future, or allowing it to be a bit more dynamic. I don't know.
That'd be great.
Yeah. I guess we're coming up to 5 past, and there's still a lot of questions. So I don't want to take more time, especially of people online. Going to have to go to other meetings, I'm assuming. So we'll follow up those questions online. Thanks again to Lysan and Maria. That was a brilliant presentation. If I could just have a couple more slides just to highlight a couple more webinars that are coming up for your diaries. So the first one is around best practice framework for applying PBPK modeling in pediatric drug development. So that's hosted by Karen, Trevor Johnson, and Eva Gill Berglund. And that will be on Wednesday, the 19th of May. I guess there's a long list of things that they're going to cover in this, looking at application of PBPK, looking at best practices and looking at the guidelines, and essentially a summary of those guidelines and the best practices. The next question, next webinar to also put in your diaries is our scientific webinar series 1 in June, and David Taft is going to be looking at a case study looking at 1A2 modulation, yeah, please put that in your diaries as well. That's 30th of June. Thank you everyone, and thanks again to our fantastic speakers today, and take care.
Thank you. Bye.
Bye.
Thank you. Our pleasure. Bye.